am Dr. Swati Sharma from DRCDU. Today we would be discussing the sterilization techniques which are used in microbiology. We first of all need to understand what is microbiology. The term microbiology is coined from the Greek word which consists of two words. Micros means small and bios which means life. So anything which is living and need to be seen under the microscope comes under the term microbiology. So it is the study of the organisms which are minute in size. These organisms can be unicellular, that means have only single cell, or they can be multicellular, forming the cell colony, or acellular, which means lacking the true cells such as viruses. So all the organisms which are so small that they cannot be seen from the naked eyes and need to be seen under the microscope are termed as microorganism, and the field which study these microorganisms is termed as microbiology. The field of microbiology encompasses numerous subdisciplines such as virology, parasitology, mycology and bacteriology. So when you decide to study the microbiology, you have to culture these microbes, and for culturing these microbes there are certain techniques which one need to follow, and these techniques are known as sterilization techniques. When we know that most type of the microbes are unknown, and it is still estimated that we know only 1% of the microbial species on the earth, and all the microbes which are around us everywhere in the air, water, soil are still unknown. And if you see, an average gram of the soil contains 1 billion microbes, which represent probably the several thousand species. So how can we study them? We have to first culture them to study them, and there are two different types of sterilization methods. One is physical and other is chemical, and then these methods are further divided into various subclasses, such as the physical is further divided into heat sterilization, which can be dry heat or wet heat. Then with the help of the radiations, these radiations can be ionizing radiations or non-ionization radiations. Then use of filters such as HEPA filters. Nowadays you guys must be hearing that we should have air purifiers at our home because of the pollution. So these air purifiers contain HEPA filters. And then we have the second class of sterilization, which is chemical sterilization. It involves the treatment of the culture surface with certain chemical agents such as alcohol, formaldehyde, etc. And then there is a combination of the physical and chemical method, which is termed as physiochemical method. So let's first try to understand what are the physical methods of sterilization. First we are talking about heat sterilization. So here, as I told you, we have again two subtypes, wet sterilization and dry heat sterilization. In wet heat sterilization, we use the moist heat or the heat having the water vapors to sterilize the equipments or the instruments which are needed for the microbial culture. In dry heat, we use the flames without any moisture in order to disinfect the surface or certain instrument. In dry heating, we use the spirit lamp for making the surface red hot. The second method of physical sterilization is filtration. So filtration can be of liquids and gases. There are different types of the filters. For filtering of the gases, we have HEPA filter. So all the microbial work is done in the hood, which is known as a laminar hood, and in the top of the laminar hood there is the HEPA filter, which prevents the entry of microorganism inside the hood, so that the sterile conditions are prepared in the hood. And the second filtration is filtration of the liquids, so that the liquids in which the microbes are being cultured are sterile. So for them we use syringe filters. The third method of physical sterilization is irradiation, using different kind of rays such as UV rays, gamma rays and X rays. UV rays are highly lethal to the microorganism because they break the DNA, and microorganisms are the prokaryotes. They lack the DNA repair mechanism and hence they cannot survive. So the longer exposure of the UV leads to death of the microorganism. There are certain UV is an example of non-ionizing radiation. There are certain ionizing radiations also, such as gamma radiation. Other physical methods involve sound, which is sonic wave or ultrasound, pressure, and the use of direct sunlight or solar disinfection, though last three methods are less commonly used in the labs. The chemical method which is most commonly used is the use of 70% alcohol. So why 70% alcohol is used? Because first, it's a dehydrating agent. It dehydrates the cells by dissolving the membrane lipids and also denature the biomolecules, which includes proteins, and thus it kills the organism. It is effective against most of the bacteria and fungi and also the viruses. So it is used for the sterilization of the working surfaces, palms, etc. But why only 70%? Why not 50% or 90%? Because 90% of alcohol will evaporate very easily and 50% will not degrade the membranes effectively. That is why 70% alcohol is always the choice. And you must have noticed when there was COVID breakdown, everyone was suggested to wash your hands or use 70% alcohol. All the sterilization that we use is 70% of alcohol. The commercial sterilizer that are present, they are nothing but 70% alcohol. The other method is UV radiation. So it's also a sterilization method which uses the light which falls in the range of ultraviolet, and it's the sufficiently short wavelength which breaks down the microorganism because it intercalates in the DNA. It produces the breaks in the DNA. This light is also used in various applications such as in food industry, air and water purification. You guys must have seen the water purification at your home. RO purifiers, they also have UV light in it because it breaks down the microbes which are present in the water, hence purifying the water. So UV will break the molecular bonds within the microorganismal DNA. Thus it produces thymine dimers in their DNA, thus destroy them. It renders them harmless or prohibit their growth and reproduction. Short range UV is used usually and it is termed as UVC, which is ultraviolet short range, and it is germicidal. Its wavelength is 254 nanometers. The next step is dry heating. So for dry heating, two things are most crucial. First is the time for which you are heating your instruments or the equipments, and second is the temperature. So the proper time and temperature for dry heat sterilization is 160 degrees Celsius for 2 hours, or you can increase the temperature to 170 degrees Celsius for 1 hour. Instrument should be dry before sterilization since water will interfere with the process. Dry heat will destroy the microorganism by causing the coagulation or degradation of their proteins. The stream sterilization will use the moist heat or the wet heat. Autoclaving is such a technique in which you use stream sterilization. So autoclaving at high pressure, which is 15 psi for at 121 degrees Celsius for 10 to 20 minutes is sufficient to kill all the microorganism. And it is the most commonly used techniques in the labs. We have different type of the autoclaves which are used for stream sterilization. Filters are again another physical method. They are the clear liquids that would be damaged by the heat. So we cannot use the heat sterilization method for those liquids, or irradiation by the UV light or chemical sterilization. So they need to be sterilized by different way, and one of such simplest way is filter. So you use membrane filters in order to pass clear liquids from them, which will prevent the entry of the microbes, as the microbes would be trapped in the sieves of the filter. The next technique is pasteurization. It is heating at 71 degrees Celsius for 15 seconds. which is followed by rapid cooling, such that the microbes will feel heat shock, because first they are heated for 71 degrees Celsius for 15 seconds and then there is a rapid cooling. So, the microbial cell wall will disintegrate and microbes will die because they are continuously feeling the heat shock, and this is called as flash pasteurization because we are giving the flash of the heat. This I explained to you earlier that why 70% alcohol, not 90% or 50%, because pure alcohol will coagulate the protein in the contact and it would be harmful for your own skin also, and it will evaporate very fast. And the less percentage of the alcohol such as 50 and 60, it will not be able to disintegrate the membrane properly. Hence, 70% ethanol has been found to be the most effective in killing the microbes. Higher or lower concentration are not so effective in killing the microbes. If you use 90% or absolute, it will evaporate fast and may not be much effective. So, this is the structure of the laminar hood. This is the workspace for all the microbiology experiments, because for the microbiology experiment you want to culture only the species of interest. You do not want any other contamination. So, you use a sterile space. The function of this laminar hood is to provide that sterile space. The top of the hood have HEPA filter, which is high efficiency purification air filter. It will trap the microorganism and the air will flow through it. The lower or the steel surface is your workbench, which one wipes with 70% alcohol before starting the experiments. Thus, it is a closed container or the closed space for conducting all the experiments. This is the autoclave. The autoclave is used for the steam sterilization or the wet sterilization. As you can see, it will have a meter above it. So, the meter will give you the reading of the pressure and the temperature, and you will maintain the high pressure, which is 121 psi, for 15 to 20 minutes, so that the temperature rises up to 121 degrees Celsius and all the microorganisms will die at that temperature. These are the different variations of the autoclave. Once you have your lab, you have to decide which type of the autoclave you want to use. If you have only few instruments which you need to sterile, you will use a pressure cooker type autoclave. So, it is a very small autoclave and it can be used for few instruments. Then you have the vertical autoclave, which you use for the larger experiments. When you have large instruments or when you have more cultures to be autoclaved. Then you have one horizontal autoclave, which is mostly used in the hospitals, because before hospital में जो भी waste generate होता है, it is rich in the biological samples and it can attract the microbial growth. So, before it can be discarded, it need to be disinfected. So, before discarding, the disinfection is done in the horizontal autoclave. So, these are the different type of autoclaves depending upon their uses. This is the standard autoclave. All different type of the autoclaves will have all these instruments in them. They will have a pressure meter from where you can see the pressure. They will have a inlet from which you will put the water. They will have an outlet for the steam. They will have a temperature reading. They will have a stand and they will have a container on which you will place all the things that need to be autoclaved, and it will have an outer body, which is the autoclavable body, which acts like a container. So, this is the basic mechanism of the airflow in the autoclave. Once it is completely sealed, the pressure causes the temperature to rise, and that rise in the temperature causes the water beneath to vaporize, and that heat from those water vapors, it will kill the microbes and thus disinfect all the things. So, these are the categories depending upon the temperature. So, if you want to maintain the temperature of 115 to 118 degrees Celsius, then the pressure has to be 10 psi and the heating time should be 30 minutes. If you want the temperature to be 121 to 124, the pressure has to be 15 psi, and you have to maintain that temperature and pressure for 15 minutes for the complete degradation of the microbes. If the temperature is 126 to 129, then the pressure has to be 20 and the time 10 minutes. And if the temperature is 135 to 138, the steam pressure has to be again 30 and the time will be reduced to 3 minutes. So, all this depends upon your design of experiment. For example, if you want to disinfect a microbial culture which you suspect can have thermophilus bacteria, then you will use the higher temperature, that is 135 to 138, because these bacteria, they can survive under high temperature as well. So, the lower temperature such as 115 or 121 will not be sufficient to kill those thermophilus bacteria. In that case, you will use the highest temperature, which is 135 to 138. But in the routine cases, we usually use the temperature which is 121 and pressure 15, with the holding time which varies from 15 to 20 minutes. And now we have the other instrument which is called the incubator. Once everything is sterilized, you need to store it. So, you store in an instrument which is called incubator. So, incubator provides the space for the microorganism to grow. Once you culture your microorganism either in the solid media or in the liquid media, you put them in the incubator, because the incubator will provide a steady temperature and the agitation. Right hand side, there is an incubator with the agitator. You can see the flask. So, it can shake the flask for the continuous mixing of the culture. All microorganisms, they have a optimum pH and optimum temperature at which they grow. So, the purpose of the incubator is to provide the optimum temperature for the microbial growth along with providing the sterile surface. These incubators are always cleaned and they are sterile. They maintain the temperature and humidity for the proper growth of the microorganism. And also they provide the shaking incubator provides the optimum shaking to the media, so that there is continuous mixing of the culture. And now moving to the heat sterilization. As you can see, this is the dry heat, because we are using the flame directly to make the rod red hot. So, once the rods are red hot, they are cooled down and they are considered fully sterile. So, these are the different type of the rods or the tubes which are used for inoculating the bacterial colonies. So, on the top you can see the metal rods. They can be used multiple times after the heat sterilization. At the bottom you can see the plastic rods. They have the loop to pick the single colony, and these plastic rods, they are never heat sterilized. They are only one time used. After using, they are disposed of. Only the metal ones are heat sterilized and can be used multiple times. Then you have the loops and the T-rods. The function of the loops and the T-rods is again the streaking or the inoculation of the bacterial colonies in the solid agar plates. So, the glass and the metal loops can be heated and made red hot and can be again used and reused. With the plastic T and the L loops, they cannot be reused. They are used only once and then disposed of. Now, let us consider different methods of the streaking. Because we are studying microbiology, we need to culture the microorganism, and for culturing the microorganism, there are different ways. Streaking is one of the way of culturing the microorganism on the solid agar plates. So, in streaking, first a single colony, pure colony is picked up from the stalk. As you can see on the left hand side, a loop is used to pick up the single colony from the already cultured plate, and then you put that colony in the agar plate and you do the streaking in the zigzag manner. As you follow these three slides, the streaking is first done on the top side, then the left and the right, making sure that the entire surface area of the plate is covered. And this is how the lawning is done. The second method is lawning. In lawning, you do not use the metal tube. you use the glass rods. So, glass rods are first heated and turned red hot to sterilize them. Then they are cooled down so that they do not kill the microorganism. Then the bacterial colony is picked from the already cultured bacterial plate, and then the rod is moved into the circular motion in the solid agar plate so that the more microbes can be cultured, and then these plates are put in the incubator for overnight so that the bacteria can grow. The temperature is maintained at 37 degrees Celsius, humidity is maintained from 55 to 60 degrees Celsius, and the growth is observed the next day. But before you start all these experiments, you have to make sure that your hands are clean. We are doing sterilization techniques today. So, first thing you sterilize is your hands. So, there is a proper way to disinfect your hands before you start any microbiological experiment. First you rub the top portion, then you rub in between, and then you rub in between the thumb and your index finger, and all this is done with 70% ethanol or alcohol. So, that is the basic disinfectant that you use for your hand, so that your hands are clean and they are devoid of all microbial growth. And once you disinfect your hands, you wear the gloves and again you wipe your gloved hands with 70% ethanol to avoid any kind of the microbial contamination. So, this is your sterilization. This is how you take it in the large quantity so that it can cover the entire surface area of your hands. Now, let us move to another physical method, which is the use of the radiation. Let us first consider the non-ioning radiation, which is UV radiation. So, UV irradiation also causes the sterilization. I explained to you the mechanism that it breaks the thymine bond and makes DNA unstable. So, for the larger spaces such as the hospitals, such as the ICU units and the cancer wards, they all are lined with the UV lights and they are periodically sterilized by the use of UV lights. So, for the sterilization with the UV light, you have to make sure that you are switching on the UV light when no one is present, because this UV light can be lethal to all the living beings including humans. So, once the UV light is on, there should be no individual present in that room, and the light is switched on for minimum 15 to 30 minutes so that all the microorganisms which are present in that room or the large surface area are exposed to UV light, and UV light can damage their DNA and hence can stop their growth and replication and reproduction. So, for the larger units such as the multi-bed ICUs or the immunocompetent cancer patient wards, the UV light is switched on for overnight, giving it enough time to sterilize the entire space. The other kind of lights that can be used is gamma lights or the gamma radiations. Gamma radiations are ionizing radiations. They ionize the molecules which are present in the microbial cells and thus causing their disintegration and breakdown. The gamma irradiations are used for the smaller instruments. So, the chamber that you are seeing on the top is a closed chamber in which you can put your all the instruments, close the chamber and then switch on the irradiation. All the instruments which are present, they will be exposed to the radiation for 15 to 30 minutes and they would be disinfected. Now, let us move to the other part of the sterilization technique, that is by the physical method, the filtration sterilization. Once you know that no other kind of sterilization can be done on your culture like heat sterilization, because you have proteins which will be broken down because of the heat, or chemical sterilization because the chemicals can affect your culture, then you use the filter sterilization. So, there are different type of the filters which are present based upon their pore size. The pore size can vary from 0.2 micromolar to 10 micromolar. There are different kind of HEPA filters. The HEPA filters have the steel wool and the glass wool which forms a mesh like structure and makes a sieve. Then you also have different type of membrane filters. The membrane filters are also again the sieves and the candle filters for the vacuum units. So, the top picture shows the syringe filter. It is a smaller filter, and you can see the larger molecules can pass through it, but the smaller ones are trapped, and hence the filters can be used. So, this is the common HEPA filter which we use. It has the steel wool and the glass wool. So, let us conclude our session on the sterilization techniques by this. Thank you.